Hepatitis virus is a viral hepatitis is a systemic disease with a primary infection in liver. There are six hepatitis viruses, that is hepatitis A, B, C, D, E, and G. Hepatitis virus is a DNA virus, while other A, C, D, E, G contains RNA genome. Hepatitis A virus is a HAV, infectious hepatitis. Infectious hepatitis is a subacute disease occurring mainly in children and young adults. Hepatitis A virus enters the body by oral route through the contaminated food, water, or milk. Morphology: Hepatitis A virus is 27 nanometer non-enveloped single stranded RNA viruses with an isosaccharide symmetry. Resistance: the virus is resistant to inactivation by heat at 60 degrees centigrade for 1 hour, either and to pH 3. It survives at 4 degrees centigrade or below. It is destroyed by autoclaving, boiling for 5 minutes, formaldehyde, and by chlorine at 10 to 15 ppm for 30 minutes. Laboratory diagnosis: demonstration of virus. The immunoelectron microscopy. The virus can be visualized by immunoelectron microscope in feces. The enzyme link immunosorbent assay. Hepatitis A virus can be detected in feces. Isolation: the virus can be grown in human and in simian cell culture. It is not possible to grow them routinely from feces of patient. Detection of antibody: because of difficulty in isolation and detection of virus, diagnosis depends upon the demonstration of a specific IgM antibody to hepatitis A virus in blood. The IgM antibody is diagnostic of a recent infection, and while IgG antibody is useful indicator of a past infection as it persists for many years. ELISA kits for detecting IgM and IgG antibody are available. Biochemical test: liver function test such as a serum alanine aminotransferase and bilirubin level supplement the diagnosis. Prophylaxis: improved sanitation, prevention of fecal contamination of food and water. Formalin inactivated vaccines containing hepatitis A virus grown in human diploid cell culture is available. A full course containing of 2 intramuscular injections of vaccine protects last for 10 to 20 years. One attack of disease gives long-term protection against hepatitis A virus. Hepatitis B viruses. Hepatitis B virus differs from hepatitis A virus in various aspects. Morphology: hepatitis B virus is a complex 42 nanometer double-shelled particles. The outer surface or envelope of a virus contains hepatitis B surface antigen. It encloses an inner ecosaccharide 27 nanometer nucleic acid, which contains hepatitis B core antigen. The inside the core, it is genome. It is in the genome a circular double-stranded DNA and a DNA polymerase. Hepatitis B surface antigen, or previously named as Australian antigen. Electron microscopy of serum of hepatitis B patient shows 3 types of particles. The most abundant form is spherical particles 22 nanometer in diameter, and the second type is tubular 22 nanometer in diameter particles of varying length. These two types are antigenically identical and are surface subunit of hepatitis B viruses. The third type is a double-shelled spherical structure 42 nanometer in diameter. This particle is complete hepatitis B viruses or Danny particles. Antigen structure: hepatitis B surface antigen. The surface antigen envelope protein. There are 4 antigenic type: hepatitis HBsAg, ADW, ADR, AYW, and AYR. The type AYW is predominant in Africa, Russia, and in India. The hepatitis B core antigen. It is a core nucleocapsid antigen of the viruses. It is not detectable in patients' blood. Hepatitis B envelope antigen. It appears in serum along with hepatitis B surface antigen, but it disappears within a few weeks. It is hidden antigenic component of core viral gene and antigens. The viral genome consists of 2 linear strands of DNA held in circular configuration. One of the strand, the plus strand, is incomplete, while the other is complete. Associated with the plus strand is a viral DNA polymerase. The genome has 4 genes with different regions coding from different antigens. Mode of transmission: parental, perinatal, and sexual. Hepatitis B carrier. Super carriers, they have hepatitis B envelope antigen in blood and are highly infectious. This blood contains a high titer of hepatitis B surface antigen and DNA polymerase. Very minute amount of serum or blood can transmit the infection. simple carrier. They have no hepatitis B envelope antigen and low level of hepatitis B surface antigen in blood. Hepatitis B virus and DNA polymerase are absent. They transmit the infection only when large volume of blood or serum are transferred, as in blood transfusion. They are more common type of carriers. Laboratory diagnosis. The laboratory diagnosis of hepatitis B virus infection can be carried out by the detection of hepatitis B antigen and antibody. These can be detected by sensitive tests like ELISA and radio immunization. Hepatitis B core antigen is not detectable in serum but can be demonstrated in liver cells by immunofluorescence. The sequence of appearance of viral markers, hepatitis B antigen and antibody in blood is important in diagnosis. Hepatitis B viral DNA level can also be detected in serum by polymerase chain reactions. Prophylaxis, general preventive measures, avoid use of unsterile needles, syringes and other materials. Screening of hepatitis B surface antigen, hepatitis E antigen in blood donors. Health education, immunization, passive immunization. Hepatitis B immunoglobulin can be given intramuscularly as early as possible after an accidental exposure to hepatitis B infections. Active immunity, plasma derivative vaccines. Prepare by purifying 22 nanometer particles of HBsAg from plasma of healthy carriers. It is inactive with formaldehyde. Recombinant yeast hepatitis B vaccines. It is produced by the recombinant DNA in yeast in which a plasma containing the gene of HBsAg has been incorporated. The HBsAg particles produced are extracted and purified for use as vaccines. These 3 doses of 0, 1 and 6 months are administered intramuscularly into the deltoid muscles. The recombinant Chinese hamster ovary cell hepatitis B vaccine. The CHO cells have been used for preparation of vaccines treatment. No specific antiviral treatment is available. The interferon alpha alone or in combination with other antiviral agent lamivudine and famciclovir has been beneficial in some chronic hepatitis cases. Hepatitis C virus is a 50 to 60 nanometer virus with a linear single stranded RNA of positive polarity and surrounded by an envelope. It has not been grown in culture but has been cloned in E. coli. Mode of infection: the parental by contaminated needles and syringes, transfusion of infected blood and blood products, sexual. Laboratory diagnosis: the detection of anti-HCV antibody by ELISA, a recombinant immunoblot assay, radio immunoblot assay, recombinant immunoblot assay can be performed to confirm the positive result obtained with a screening test such as ELISA. The viral genome HCV RNA detection by polymerase chain reactions and by immunofluorescence. Prophylaxis: screening of blood or blood products, avoidance of use of unsterile needles, syringes and other materials. Hepatitis D viruses. Hepatitis D virus is a defective virus as it is dependent on the helper functions of hepatitis B virus and its replications and expressions. Hepatitis D virus is a spherical 36 to 38 nanometer diameter RNA particle surrounded by hepatitis B surface antigen envelope. The genome is a single stranded small circular molecules of RNA. It encodes its own nucleoproteins, the delta antigen or hepatitis D antigen, but the outer envelope of hepatitis D virus is encoded by the genome of hepatitis B virus co-infections. These same cells. Hepatitis D virus is necessary for protection of hepatitis D virus. No association has been noted between hepatitis D virus and hepatocellular carcinoma. Laboratory diagnosis: the detection of a IgM anti-delta antibody in serum by ELISA or radioimmunization. Hepatitis D antigen, delta antigen, is primarily expressed in liver cell nuclei where it can be detected by immunofluorescence. Hepatitis D virus RNA can be detected by hybridization technique. Prophylaxis: no specific prophylaxis. Immunization with hepatitis B vaccine is effective because delta antigen cannot infect person immune to hepatitis B virus. Screening of blood donors of hepatitis B surface antigen will also limit blood-borne hepatitis D viruses. Hepatitis E virus. Hepatitis E virus is spherical, non-envelope and 27 to 38 nanometer diameter. It possesses a single stranded RNA genome which is surrounded by the ecosacral capsule. Mode of infection: ingestion of fecally contaminated drinking water. It could predominantly in young to middle-aged adult. Clinically, the disease resembles that of hepatitis A. Laboratory diagnosis: exclusion of hepatitis A and hepatitis B. The feces examination by electron microscope for calcivirus-like particles. The antibody detection by ELISA and Western blot assay. The detection of hepatitis E virus RNA in feces by polymerase chain reactions. Prophylaxis, improved sanitation, chlorinated water. Hepatitis G viruses. The genome of hepatitis G virus consists of a single strand RNA. The virus is transmitted parentally, sexually, and from mother to children. There is no evidence of a relationship between hepatitis G virus and hepatitis carcinoma virus. Laboratory diagnosis, reverse transcriptase polymerase chain reactions, RT-PCR antibody detections, prophylaxis. The same general prophylactic measures which are used for hepatitis B virus and hepatitis C virus.